I was just reading the latest clinical data on ketone metabolism, and it changes how we should evaluate cognitive flexibility in healthy aging adult populations. Many people experience distinct dips in mental energy and focus during the day, assuming it is purely a result of insufficient calorie intake as we navigate healthy aging during daily cognitive tasks. We commonly treat these cognitive fluctuations as a simple requirement for glucose, without looking at alternative fuel pathways within our neural tissues in our daily lives to protect our brain structure. However, neuroscience suggests that cognitive steadiness depends partly on the brain's flexibility to switch between glucose and ketone body metabolic pathways within neural tissues as we navigate healthy aging. Imagine a system where your brain cells can smoothly transition to a backup fuel source when glucose availability falls, maintaining network performance under these metabolic conditions in our daily lives. We are exploring how ketone body utilization supports neural network stability, helping to reframe cognitive stamina from simple glucose delivery to substrate flexibility to support cognitive endurance within neural tissues. When metabolic flexibility declines with age, neurons struggle to maintain consistent electrical activity during periods of high demand or low glucose levels as we age over time under these metabolic conditions. If we address energy dips only by consuming simple sugars, we trigger rapid insulin fluctuations that further disrupt central metabolic signaling pathways in healthy adult populations to support cognitive endurance. The stakes are high because reduced brain metabolic flexibility is associated with diminished synaptic plasticity and accelerated cognitive aging over the lifespan for long-term health span as we age over time. Understanding the physical pathways of ketone transport allows us to design practical, low-risk protocols that support our brain's metabolic resilience naturally during daily cognitive tasks in healthy adult populations. The problem is that typical modern lifestyles maintain a constant state of glucose reliance, minimizing the cellular pathways that utilize alternative substrates to protect our brain structure for long-term health spans. By identifying the specific biological mechanisms of ketone metabolism, we can establish better daily frameworks to support long-term cognitive longevity and focus as we navigate healthy aging during daily cognitive tasks. To understand this metabolic process, we must define ketone bodies, which are water-soluble molecules produced by the liver from fatty acids during fasting in our daily lives to protect our brain structure. The primary ketone bodies utilized by brain tissues are beta-hydroxybutyrate and acetoacetate, which serve as highly efficient alternative energy substrates for cells within neural tissues as we navigate healthy aging. Under normal dietary conditions, the brain relies almost exclusively on glucose, which is transported across the blood-brain barrier via specialized glucose transporters under these metabolic conditions in our daily lives. However, when glucose levels decrease or circulating ketones rise, the brain increases transport of these alternative fuels via monocarboxylate transporters and capillaries to support cognitive endurance within neural tissues. Once inside, ketones are converted directly into acetyl-CoA within the mitochondria of neurons and glial cells, bypassing the initial glycolysis steps as we age over time under these metabolic conditions. This entry into the citric acid cycle allows ketones to generate ATP, the cell's energy currency, with high thermodynamic efficiency in the tissue in healthy adult populations. Importantly, ketone metabolism produces fewer reactive oxygen species per molecule of ATP generated compared to standard glucose utilization in the cells for long-term health span. During healthy aging, the expression of both glucose and monocarboxylate transporters can experience gradual shifts, affecting substrate availability to active networks during daily cognitive tasks. As we age, the brain's capacity to transport and utilize glucose can decline, a state sometimes referred to as glucose hypometabolism to protect our brain structure. This hypometabolism creates a local energy deficit, especially during demanding cognitive tasks that require rapid, synchronized activity across different cortical regions as we navigate healthy aging. When glucose transport is reduced, but monocarboxylate transporters remain functional, ketones can serve as a vital backup fuel source for neurons in our daily lives. By supplying acetyl-CoA directly to mitochondria, ketones sustain the cellular ATP levels required to maintain the resting potential of neuronal membranes within neural tissues. This maintenance is essential for supporting synaptic transmission and preventing the excitotoxic injury associated with cellular energy failure in the brain under these metabolic conditions. Beta-hydroxybutyrate also acts as a signaling molecule, inhibiting histone deacetylases to promote the expression of brain-derived neurotrophic factor in the hippocampus to support cognitive endurance. This epigenetic signaling supports neuroplasticity, illustrating that ketones serve both as an efficient fuel substrate and a regulator of connection density as we age over time. Furthermore, utilizing ketones reduces the accumulation of metabolic waste products by promoting mitochondrial efficiency and cellular autophagy pathways in glial cells in healthy adult populations. When autophagy is active, damaged organelles are cleared, lowering the level of local cellular stress that impairs oligodendrocyte and myelin maintenance for long-term health span. This dual metabolic and signaling role of ketones helps sustain executive function during periods when glucose availability is temporarily reduced in daily life during daily cognitive tasks. The transition between fuel sources, however, requires the active maintenance of the enzymatic pathways responsible for ketogenesis and ketone oxidation in tissues to protect our brain structure. In a state of constant carbohydrate exposure, the enzymes required to utilize ketones are downregulated, limiting the brain's immediate metabolic flexibility as we navigate healthy aging. This downregulation prevents the neurovascular unit from utilizing alternative energy substrates when local glucose delivery is mismatched to high neural demand in our daily lives. The overall endurance of our cognitive networks is therefore closely tied to our capacity to activate these alternative metabolic pathways periodically within neural tissues. We must examine the boundaries of this metabolic flexibility model to design safe, evidence-bounded daily protocols for healthy cognitive aging support under these metabolic conditions. One limitation of ketone research is that the cognitive effects of ketosis vary widely among individuals due to genetic and metabolic differences to support cognitive endurance. We must interpret clinical data on ketogenic protocols cautiously, as long-term compliance is challenging and metabolic baselines differ significantly among subjects as we age over time. In some cohorts, brief periods of nutritional ketosis are associated with improved processing speed, though a universal protocol is not established in healthy adult populations. We must also recognize that ketone metabolism is not a standalone solution for cognitive health, as sleep and vascular health are critical for long-term health span. The population boundaries of current human studies are narrow, and we lack long-term clinical trials evaluating mild ketosis in healthy aging during daily cognitive tasks. Because we cannot directly observe real-time ATP production in living human brains, we rely on indirect neuroimaging and serum ketone markers to protect our brain structure. Therefore, we analyze ketone body metabolism as a significant contributing pathway in cognitive resilience, rather than a definitive treatment for decline as we navigate healthy aging. Answering our opening question, cognitive energy dips are often a fuel flexibility issue rather than a simple lack of overall energy resources in our daily lives. By understanding this physical mechanism, we can reframe our habits, focusing on supporting metabolic flexibility to maintain stable brain network performance within neural tissues. When the capacity to utilize alternative fuels is preserved, neurons maintain stable ATP levels, supporting synaptic transmission and executive focus during work under these metabolic conditions. The practical challenge is encouraging the activation of these monocarboxylate transport pathways through low-risk daily behavioral and nutritional protocols as we age to support cognitive endurance. While we cannot modify all age-related shifts in glucose transport, we can take steps to promote the availability of alternative substrates as we age over time. Safe, structured periods of fasting or moderate carbohydrate restriction are associated with increased liver ketone production and enhanced central transport markers in healthy adult populations. Regular physical exercise promotes metabolic flexibility, helping the body switch between substrates more efficiently during both rest and physical exertion for long-term health span. These metabolic habits do not target memory storage directly, but they support the cellular energy infrastructure required for optimal synaptic performance during daily cognitive tasks. Supporting brain metabolic flexibility requires a unified focus on dietary consistency, regular movement, and proper sleep to optimize these vital pathways to protect our brain structure. To apply these insights, discuss your metabolic health and potential dietary patterns with a qualified healthcare professional during routine clinical checkups as we navigate healthy aging. Incorporate consistent physical exercise into your weekly schedule to support systemic metabolic flexibility and promote cardiovascular health naturally and safely in our daily lives. Consider structuring your daily meals to allow a consistent overnight fasting window of 12 to 14 hours to support natural ketogenesis within neural tissues. Consult a physician to monitor metabolic biomarkers like fasting glucose, insulin, and lipid panels, as systemic health supports brain function under these metabolic conditions. Avoid self-prescribing highly restrictive ketogenic diets or exogenous ketone supplements without guidance, as individual tolerance and cardiovascular profiles vary widely to support cognitive endurance. Please remember that this analysis is intended for educational purposes and should not replace individualized clinical evaluation or specific medical advice as we age over time. Focus on establishing simple, low-risk daily habits that support overall metabolic efficiency and reduce systemic cellular stress naturally and safely in healthy adult populations. Track your daily activity, fasting patterns, and cognitive focus to build a personalized, evidence-bounded framework that supports your long-term health span. Optimize the protocol, protect your brain, and I'll see you in the next data set.